Thank you. So I want to thank you all for the opportunity to share some of the exciting things that are going on at Unigene and some of the developments we've made on our technology in the recent years. Can you go to the next slide? So just a quick overview on Unigene. We really believe that we are a leader in the design, development, and delivery of peptide-based therapeutics. Unigene was founded in 1980 by two brothers from the Roche Molecular Institute of Biology. And these two scientists were really big on developing new technologies and focusing them on internal programs. It wasn't until recently, in 2010, under new management, where Unigene started taking these new technologies, and especially our oral delivery technology, and trying to partner that out. But for the course of this topic, I'd like to go into some of the results we've seen on the oral bioavailability of peptides and the developments we've made on our technology. Next slide, please. So in 2012, Unigene, we've added 7 new feasibility studies to our portfolio, totaling 14 since we started this new strategy last year. So we're really excited with that number. Actually, 2 big pharma partners have come back with multiple peptide candidates. And we really have been learning about our technology and developing it further with varying the excipients and coats and everything. And we were able to achieve 3 times greater increased bioavailability compared to our oral PTH clinical trial material for the study we completed last year. Cara Therapeutics was one of our first partners that came on our new strategy. They successfully completed a feasibility study in the beginning of 2011, and were able to go into phase 1 trials the end of 2011. We were actually very happy with that study. They were able to achieve 16% bioavailability with our technology. Moving on to stealth peptides, they again saw very favorable results in a feasibility study with us, and they're looking to move into the clinic later this year. And then actually last week we just announced that Terex Pharmaceuticals completed a feasibility study with us. They saw very favorable bioavailability. Actually, it was greater than their subcutaneous formulation. And we announced a worldwide license agreement with them for our technology for that molecule. Next slide, please. Now, I'm speaking to the choir here. We all know that peptides are very difficult to deliver orally, very susceptible to protease degradation, low permeability across the intestines, and a lot of inter- and intra-subject variability, not to mention patient compliance with taking it on food and too much or too little water. With our Peptelligence technology, we really believe that we've tackled many of these issues, minimizing food effect, reducing peptide degradation. We don't modify the peptide at all. It's all in the formulation, and it all happens in a very localized microenvironment and not across the entire gut. Next slide, please. And our technology is really proving itself out in the clinic. We have a positive phase 3 oral calcitonin product, looking to file with the FDA later this year, early next year. As I mentioned, the positive phase 2 oral PTH study, and then our partner Cara, who completed their study end of last year. And as I mentioned, our 14 ongoing or completed feasibility studies. Next slide, please. So how does our technology work? We use an enteric-coated tablet to bypass the stomach. Once in the duodenum, as pH rises, the enteric coat dissolves, releasing our 2 main excipients. The first being an organic acid that we use as a protease inhibitor, and the second is an acyl carnitine that we use as a permeation enhancer. The acyl carnitine opens the tight junctions between the cells to allow for paracellular transport, and we find we can get a very reproducible PK profile, very sharp PK profile, with very little bioavailability, with being expected to oral delivery of a peptide. Next slide, please. This is just a schematic of our standard formulation. Outside is the enteric coat. Underneath that, we use a subcoat, and that's to allow the enteric coat to dissolve completely because we don't want any of the excipients or API to leach out. So there's been quite a few developments, and especially this year alone, optimizing those coats to achieve the greatest bioavailability. And one thing you'll notice is that the organic acid is in coated beads. That's to prevent the peptide and the acid from commingling during storage. So we've really been able to achieve 9 months room temperature stability. And then on the bottom is a picture of our standard tablet compared to a daily multivitamin. So it is a relatively small tablet. Next slide, please. Here's some data from our sponsored studies. When we do a feasibility study, we tested in first a rat model and then a dog model. Here's a range from those studies. In the rat model, unformulated compared to formulated is quite a huge increase, along with the dog studies as well with our formulation. Next slide, please. Here's a graph from our phase 1 PTH study. It's to show inter- and intra-subject variability across the subjects in that study in 2 different time points. And we are able to achieve a very reproducible PK profile, a nice sharp profile, which for PTH is required for efficacy. Next slide, please. And how we do our feasibility studies is as I mentioned, a two animal model. First, we do an intraduodenal injection of a liquid formulation, and that's to test if we are able to deliver the peptide orally. Next, we'll move into a solid dosage form in a dog model, and that will really give us a true indication of the bioavailability levels we'll see in man. Next slide, please. As I mentioned before, we are building sort of a really robust portfolio, and our technology is a platform technology. It can be applied to a large variety of therapeutic areas and peptides. And this just shows some of the partners that we're able to announce and and some of the indications that we're going after. Next slide, please. And just to highlight our partners. Are there any questions?
Questions for Justin? I've got one right off the top.
Okay.
What is the maximum payload of peptide that you can deliver in one pill?
We haven't really tested the upper limits. We really believe it's around 100 to 150 milligrams.
100 to 150 milligrams?
Yes.
Excellent. And are there any barriers to entry beyond that?
Yeah, I mean, it does have to, the size of the peptide, we really believe our range is right around 50 to 60 amino acids. If the peptide has a propensity to aggregate or the solubility really plays a factor. So it really is peptide specific on the levels of bioavailability we'll see.
Okay. Please join me in thanking Justin.
Thank you.
Okay, next pleasure.